Good morning, everybody. Welcome back to to the resumption of our lecture series again. Let's give a couple more minutes for the student to come back. I see about 12 students now, so just to give maybe 1 minute or two. This is the first class after the winter storm. Maybe we won't have a full class today. So I hope everybody is doing all right. So did you lose power and water?
Oh yeah, for like 4 days.
4 days. Oh my God. Yeah. I didn't have water because my pipes were broken, burst. Then I always have water in my neighborhood, but I didn't have for a couple of days, and I don't have power for 2 days or so. That wasn't very bad. So just remember to prepare yourself for the next disaster. It seems whatever we have, no matter it's a hurricane or this winter storm, the city is not very well prepared. Texas is not very well prepared. We always lose power. They always get flooded. I don't know why. It happens again and again. In the past, I feel it is okay, but after this time, I start to feel, well, maybe they should do something to improve the infrastructure, so we don't get hit badly every time. Whatever happens, we get hit really badly. I hope everybody is doing all right now. I hope you have power, you have clean water. At least in my neighborhood, it seems everything is kind of back to normal. So we have to adjust our schedule. Here, showing on this slide, I hope everybody can see my shared screen. We lost 1 week. So we get to push some of the content to the subsequent weeks. So then one of the major changes is the presentation schedule. Group 3 was scheduled to present last Friday, but apparently you couldn't. So I moved you to this Friday. It's February 26th. Okay, I hope you guys will prepare for this presentation. And then next week, we're gonna have 2 student-led presentations. One's from group 5, the other is from group 6. Because as you know, this is a way of giving you grade for this course. So everybody must have the fair share. That's 2 presentations. I can't allow just one group to give 1 instead of 2, while the others are all having 2 opportunities. So next week, we're going to have 2 presentations on Wednesday and Friday. So this week, I'm going to deliver 2 lectures. And next week, I'm going to have 1 lecture, while the other 2 times are saved for student-led discussion. So I uploaded this to the class materials in Teams, so you can go there. But the changes are mainly for group 3, 5, not 6, because group 6 was scheduled to present on March 5th. That's according to your original schedule. Okay. So if the 2 groups, group 3 and group 5, have questions, you can ask me now or later. The material for group 3 has been uploaded already, but for group 5, you can expect to see it around Wednesday, your presentation material. So today we're going to continue to discuss stem cell biology. The content is about signaling pathways. So this lecture is going to be very technical. I don't I don't expect that everybody can memorize what I'm going to say. So just to try to learn as much as you can, and maybe summarize the common features of different pathways. So you can have a general principle, a general idea of how signaling pathways works. So I'm going to describe 5 signaling pathways. These pathways apparently are not going to be restricted to stem cells. So these are, major signaling pathways for many processes. For growth, for metabolism, for many things. They all use the same pathways, the same set of pathways. So today I'm going to give you 5 of them. So apparently they're involved in regulating stem cell proliferation or differentiation. That's why we include it in here. You can see the contents in chapter 4 of the textbook. So I'll start with the WNT beta-catenin pathway. So this is one of the most famous and one of the, the one that gets established in early days. So when the beta-catenin pathway. For most of the pathways we're going to discuss, it involves some kind of a extracellular ligand and receptor. The receptor is on the cell membrane. Usually it's a protein or protein complex. And then the ligand is either short peptide or it is another protein. It could be secreted or it is anchored in the adjacent cell. So 2 types of situation. It could be, secreted. It's soluble, floating around outside of the cell membrane, or it could be anchored to adjacent cells. So in this case, WNT is the ligand. The ligand is not anchored anywhere. It's a secreted protein by other cells. So this pathway involves a beta-catenin degradation complex. That complex is mainly formed by 3 proteins. One is GSK3 beta, the other is called APC, while the third is called axin. So these 3 proteins usually will bind to beta-catenin and phosphorylate beta-catenin. So in this slide, we don't see the phosphorylation, but the function of this complex is to bind to beta-catenin and phosphorylate beta-catenin. So once beta-catenin is phosphorylated, it will undergo degradation by ubiquitination. Ubiquitination is a kind of protein degradation pathway. Once the protein is marked by ubiquitin, it will go through the proteasome and eventually get degraded. So beta-catenin will undergo degradation. So this is a way to inactivate beta-catenin. So this is in the absence of WNT. So when WNT is not there, when WNT didn't bind to the cellular membrane receptor of the WNT pathway, now this degradation complex will degrade beta-catenin. But if you have WNT bind to the cellular receptor here, that's called freezed protein. Freezed is one member of the receptor. The receptor is actually quite complex. And here, it is an extremely simplified receptor. We have only freezed here. So freezed is the receptor. When WNT bind to freezed, it will recruit another protein called disheveled. A disheveled is a protein, that gets recruited to this activated frozen protein. By activated, I mean now it is bound to the ligand. So the disheveled here is recruited, and then disheveled will further recruit axin and GSK3 beta, the members of this degradation complex, to the cellular membrane. So once disheveled recruit the degradation complex to the cellular membrane, the degradation complex can no longer be formed. Okay, it usually requires 3 members, and now you have these 2 members, GSK3 beta and auxin, sequestered to the cell membrane. The degradation complex won't function properly. Therefore, beta-catenin is not free. It won't get marked by ubiquitin and get degraded. Instead, it will get accumulated in the cytoplasm. So you will see increased amount of beta-catenin. And then because of that, beta-catenin next will move into the nucleus. So once it moved into the nucleus, it will bind to a transcription factor called LEF-TCF. It's another family of protein that have 4 members. Okay, beta-catenin will bind to LEF and TCF, and this complex will trigger gene transcription. So the beta-catenin is a cofactor for a transcription factor. Transcription factor functions to bind to a specific sequence of certain genes and then trigger transcription from that genetic locus. So the gene gets expressed. Okay, so that's the extremely simplified version of my explanation of the beta-catenin pathway. Shown on the left, as shown here, went bind to frizzled, and frizzled will recruit disheveled, and disheveled will recruit GSK3 beta and the associated proteins, the so-called degradation complex. And the degradation complex now won't be able to degrade beta-catenin. So that's a negative. Okay, so this GSK3 beta will degrade beta-catenin, but disheveled will inhibit the inhibition complex. So there's a double negative here. Therefore, beta-catenin will become stabilized, and now it will move into the nucleus and trigger transcription. So you can see that this is a very good example of the signaling pathway. It usually involves a receptor, a ligand, and usually that will result in maybe a release of an inhibition event. And eventually, that major player, in this case is beta-catenin, will not get inhibited. So it can perform a certain function. So that's in most cases, signal transduction works this way. Okay, so we're going to have 5 different pathways. I hope you can try to memorize as much as you can, but most importantly, try to extract some general principles that are involved. And this pathway is super important because, as you will see in the next slide or the several slides later, it is involved in stem cell maintenance, but it's also important for carcinogenesis. A lot of cancers utilize this pathway to maintain cell proliferation for cancer cells. So you notice this protein called APC. That protein actually was described in colon cancer. It's a familial colon cancer. In some family, the colon cancer runs, unfortunately, in them. Almost everybody in the family eventually will develop colon cancer. Scientists found they have mutations in the APC gene. There is a mutation there in the APC gene. Therefore, this complex will not be functional because you have a mutation in APC. APC is a major component of that degradation complex. Now the degradation complex is non-functional, and beta-catenin will be constitutively active, always active. It moves to the nucleus and then trigger important genes that help cancer cells to proliferate. Okay, so that is it's a famous discovery in the past by one of the best cancer biologists, Bert Vogelstein, describing how colon cancer is developed. It's because of a mutation in the APC gene. But here I wanted that that knowledge to help you to understand that degradation complex is involved for the degradation of beta-catenin. Well, the binding of one ligand to the receptor will through a cascade of event, stabilize beta catenin. Okay, so that's my explanation of the signaling pathway itself. On this slide is some kind of application of that pathway, and the widely studied application of this pathway is in the intestinal crist stem cells. Shown on the very left is the crist, the crypt. So on top of that crypt, apparently that's the lining of the intestine. The cells are called the enterocyte. That's the lining of the intestine. And then going deeper into the crypt, to the bottom, you see stem cells. There are two types of stem cells. One is called LGR5 plus stem cell. The other is called a strange name, plus 4 stem cell. Plus 4 is because this cell, when you count from the bottom, it's from the very bottom of that crypt, that's the number 5th. If you count the cell number, it's the number 5th cell. That's why they call it a plus 4 cell, a strange name. But the important thing is you have two types of stem cells here. One's called LGR5 plus stem cell, the other is plus 4 stem cells. And then there is another cell type called Pannis cell. The Pannis cell is important for the maintenance of the LGR5 plus stem cell because they give signal, they give ligand to the LGR5 plus stem cells. So they're the source of Wnt. The Pannis cell will release Wnt into the intracellular domain, the extracellular domain, intercellular domain between the two cell types, Pannis cell and stem cell. The Wnt is released to that domain, and then Wnt will trigger the activation of the pathway. So here we're going to see the same thing I explained in the previous slide, but when you don't have Wnt, so you have 3 proteins here, but they are not activated. These 3 proteins are the component of the receptor, of the Wnt receptor. One's frizzled, the other is called LRP, the third is called LGR. These 3 proteins all contribute to the receptor, to become the receptor complex. So as I explained, when you don't have Wnt, you have this degradation complex that will phosphorylate beta-catenin and get beta-catenin degraded. So nothing will happen. The beta-catenin is inactivated. But when you have Wnt, so when the Wnt is released from the Pannis cell and bind to the receptors, the 3 receptors here, frizzled, LGR, and LRP, in the help of a 4th protein called RS binding. So these 4 proteins will help the recruitment of disheveled and additional associated protein from the degradation complex. This degradation, some components of the degradation complex now is recruited to the cell membrane. They no longer can perform the degradation function. Now beta-catenin will move into the nucleus and perform a transcription triggering function of the beta-catenin. So that pathway will help these cells, the LGR5 plus stem cells, to self-renew, to proliferate. Therefore, the Pannis cells are actually very important for the maintenance of the stem cell population in the intestinal crypt. Okay, so that's the function of Wnt in this setting. Maintain the stem cell population in the intestinal crypt. But look, one principle I want you to understand is the same pathway can sometimes lead to distinct outcomes. In this case, we have just explained the beta-catenin pathway. The beta-catenin pathway is known to promote self-renewal in the intestinal crypt. We just explained that. It helps the cells to proliferate there. However, in embryonic stem cells, that's the pluripotent stem cell, right? Embryonic stem cell is pluripotent. They are not adult stem cells like the stem cells in the crypt. They are pluripotent. They can give rise to all kinds of things. In embryonic stem cells, if you trigger the WNT pathway, you activate the WNT pathway, it's going to make the cells to differentiate. They don't like beta-catenin to be activated. The maintenance of self-renewal in pluripotent stem cells are done by different mechanisms. So it's quite common. So the same pathways, signaling transduction can lead to different outcomes in different biological settings, in different contexts. Then you must wonder how could that happen. So we have one kind of a theory. There are many theories how one signaling pathway can lead to two different outcomes in different cells. So here is a very common observation for different pathways. Basically, as I said, beta-catenin is a cofactor for transcription factors. Once it moved into the nucleus, it will bind to additional proteins. Eventually, beta-catenin, of course, will bind to the TCF, LEF, as shown in the previous slide. It's TCF, and TCF is the cofactor there, is the transcription factor. And here, it's just a transcription factor. TF means transcription factor, but actually it should be TCF. If you want to name it, it's TCF. Okay, so here beta-catenin, of course, will always bind to TCF, but it can bind to additional helper protein. For example, it can bind to this CBP protein. It's called crab binding protein. It can bind to this protein and help self-renewal. However, it can bind to a different protein, P300. If it binds to P300 in the nucleus, the outcome is cell differentiation instead of self-renewal. Okay, so these pathways are very complicated. So you can have additional modification, therefore, to lead to different biological outcomes. So that's concludes my explanation of the first pathway. And on this slide, I move on to the second pathway I want to explain, and this is called a hedgehog signaling pathway. So the hedgehog signaling pathway is that it involves ligand called hedgehog. So in these first two examples, the pathway is named after that ligand, right? In the first case, WNT is the ligand. Here, hedgehog is the ligand. We have three homologous immers. They're called sonic hedgehog, Indian hedgehog, and a desert hedgehog. So these names are very strange. They're mainly named by developmental biologists in early days. Then when they study lower species, then you have this very strange name. I don't know why they pick these names for it. So shown on this slide is the signal transduction of sonic hedgehog. So the hedgehog binds to 12-pass transmembrane receptor called patched. It's called the receptor here is called patched. And usually, patched is associated with another protein called smoothened. Smoothened is another protein located on the cell membrane. But without hedgehog, patched will inactivate the function of smoothen. So the patched will have an inhibitory effect on smoothen. But when you have a hedgehog binding to patched, that inhibition effect is relieved. So that's the function of hedgehog. So now, what will smoothen do? So smoothen will usually leads to activation of glee. So if now remember smoothing, the function of smoothing is relieved. At the beginning, it's inhibited by patch. Now it can perform its function. Okay. So let me rephrase this. It is quite complicated. Let me explain from the downstream first, which may help you to understand better what is going on. So we have downstream two proteins called GLE2 and GLE3. If you don't have hedgehog binding to the receptor, these two proteins, GLE2 and GLE3, will undergo limited proteasomal degradation. It's prosomal degradation is just ubiquitination and degradation, but this is limited degree of degradation. It's not totally degraded. Instead, it will result in the cleavage and removal of the C-terminal activation domain. So an activation domain will get removed. Therefore, these two proteins will become inhibitors. They are not activators of transcription, they are inhibitors of transcription. So these two proteins, GLE2 and 3, especially GLE3, will become a repressor. They move into the nucleus, but they are not activating gene transcription. They repress gene transcription. That's their function without hedgehog. But if you have hedgehog, and the hedgehog is going to bind to patched, and the patched is going to activate smoothing. Or shall I say, it will release its inhibition on smoothing. And then smoothing will help to inhibit the limited proteasome degradation of GLE2 and GLE3. Okay, so now the GLE2 and GLE3 will become activators because they don't have the limited proteasome degradation. Now they move into the cell nucleus. They will help the transcription of another protein called GLE1. So GLE1 is a family member of GLE2 and GLE3, but GLE1 doesn't have that mechanism. It's always active. So this now you have transcription of GLE1. GLE1 will amplify this signaling pathway because GLE1 will not have that repressor function. It's always an activator. So GLE1 will help to transcript additional genes. So that's the sonic hedgehog signal. This is complicated, but I want you to use the same principle to help you to, if you want to memorize this. As you can see, it's always binding to a receptor, and somehow inhibition event is removed from the major player. And in this case, the major player is GLE proteins. Okay, the GLE proteins are usually inactivated by proteasome degradation. And now, because of a hedgehog binding, that mechanism is no longer functional. Therefore, these proteins can move into the nucleus and help the transcription of other genes. And in this case, they help the transcription of GLE1. The GLE1 will amplify this signaling pathway because GLE1 doesn't have a repressor function. It's always an activator function. So that's the hedgehog signaling pathway. And here I show you one example how it is used in our body. Okay, it's widely studied. It's best known to be involved in the maintenance of neuron stem cells, NSC, neuron stem cells. The neuron stem cells, if you still recall in one of the lectures, I told you they can be derived from the so-called subventricular zone. That's a place, an anatomical place in the brain. And in that place, you can find neuron stem cells. So these cells require sonic hedgehog signal. How do you know that? Because if you do a genetic ablation of the gene, that means you remove that gene, knock out that gene that encodes sonic hedgehog. If you remove sonic hedgehog genetically, you are not going to have neuron stem cells. That will cause NSC depletion. That means you don't have neuron stem cells. In contrast, instead of remove that sonic hedgehog gene, now if you overexpress the sonic hedgehog gene, the result will be enhanced self-renewal. So you're going to see more proliferation, larger population of the neuron stem cells if you overexpress sonic hedgehog. Okay, so and additionally, eventually, here it is a little bit confusing. Sonic hedgehog is also required for the neuron stem cells to become the transient amplifying cell, the progenitor cell, called transient amplifying cell, the progenitors, and eventually give rise to the neuroblast. That's the immediate progenitor of neurons. Okay, so the neuron stem cell eventually will give rise to neurons. So why I mention this differentiation pathway is because the main source of sonic hedgehog appears to be neurons. The neuron is the terminally differentiated functional cell. So they also provide this signaling molecule sonic hedgehog. So you not only have functional neurons, but also you help to maintain the stem cell population in a very specific anatomic region called subventricular zone. Okay, so this is the application of the sonic hedgehog signaling, and I hope you just trying to get the principles and, if possible, tell how it is different from the first pathway we explained. I don't expect you to get everything, because this is super technical. Don't worry, just try to do your best, because gradually when you encounter the same concept again and again, eventually you're going to pick something up. But the important thing is, don't get yourself confused. Try to understand what the principle is, and try to memorize some names if you can, because the knowledge is always built up. It's not learned by one day. It's not possible. But don't confuse yourself. Don't mix up different pathways. In that way, in the future, when you have new pathways coming to you, or the same pathway, will be given to you in a different biological setting, you won't be able to learn if at the beginning you are totally confused. So clarity, precision is very important, even if you can't memorize everything, but the limited amount you can take, it must be correct and precise. And in the future, when you add up to it, you're going to learn more and more. So don't shut down yourself, because this kind of thing is it's not interesting, I know, but you cannot rely on checking textbooks all the time, because it's sometimes you just cannot do that. When you're up to the task, you have to know that in your mind, straightforward. So the third pathway I'm going to explain today is called notch signaling. Notch signaling is slightly different from the first two pathways I explained so far. They have 4 notch receptors, and then 5 structurally related ligands. So you have ligands called notch ligands, you have notch receptors. So the ligands are called delta like 1, 3, and 4, and then the other ligands are called jagged 1 and 2. So you have 5 different ligands for the notch signaling pathway. The difference here is neither ligands or the receptor, I should say that the ligand here is not secreted protein. So the ligand is also anchored in an additional cell, in the adjacent cell. Okay, so that's the difference between this pathway and the 2 pathways I explained so far. In the first 2 pathways, the ligand is a secreted protein. It's free floating in the extracellular domain. It just happened to encounter that receptor. But here, both party, the ligand and the receptor belongs to cell membrane. They're anchored to cell membrane. So the adjacent cell must align themselves, correctly for this to interact, for the ligand and the receptor to interact with each other. Okay, so the jagged is from another cell, while the notch is from the cell of interest, the signaling pathway that we are going to discuss. The notch is from this cell of interest. So the notch, just like other receptors, in this case, we call it notch pathway, but the notch is actually the receptor, not like in the previous two occasions. In the previous two occasions, there are ligands, but here we call it notch, but notch is a ligand. In this case, the notch will have extracellular domain and transmembrane domain and intracellular domain. So there is a cleavage called S2 cleavage. That is what happens when you have this ligand binding. When jagged or delta is bound to the notch receptor, you're going to have this cleavage called S2 cleavage. So this S2 cleavage will release the extracellular domain of the notch receptor, while you still have the transmembrane and the intracellular domain of the notch receptor. But that cleavage will trigger another cleavage, and the second cleavage is by a protein complex called secretase complex. So this secretase complex will cleave inside the membrane, therefore releasing the intracellular domain. So this is slightly different from what we have explained, because in the previous two examples, it's always ligand, receptor, and a major player there. But in this case, the major player notch is a transmembrane protein. It has extracellular domain, transmembrane domain, and intracellular domain, but itself moves into the nucleus. It's simpler, right? It's simpler. It doesn't involve a third party, a major player. So notch, the intracellular domain of notch, also called NICD, is the notch intracellular domain. It will move into the nucleus. Inside the nucleus, it's going to bind up to an additional protein called CSL. CSL is a DNA binding protein, just like in the previous examples, like TCF. It's a DNA binding protein, while the intracellular domain of notch will bind to CSL and activate transcription. Without notch, the intracellular domain of notch, this CSL actually performs a repressor function. It will repress the transcription from that gene. And now with the intracellular domain of notch, it gets converted to an activator. The genes are transcribed by this mechanism are called Hess and HRT families of proteins, among many proteins that are subjected to this regulation. Okay, so when you learn things and try to tell what's common, what's the general principle, and what's different. Apparently, notch is kind of different from the other pathways we have described so far. So here, I want to tell you another general principle that's signaling pathways cooperate with each other. So we are going to use the same example we explained for WNT. That's the intestinal crypt. So you have the enterocyte and you have stem cells at the bottom of the crypt. You have two types of stem cells. And then the orange cell are the progenitors or the transient amplifying cells. Well, if you move to the top, they become the lining of the intestine, they become functional, terminally differentiated cell. So if you observe the signaling molecules here, you will know that WNT the level of WNT will drop. If you move from the bottom to the surface, WNT signaling will drop because the WNT ligand is secreted by the Paneth cell. The Paneth cell is located at the bottom, and of course the gradient will become lower wind if you move from the bottom to the top. But notch is, I don't think that the source of a notch has been identified. I'm not so sure about that, but notch is kind of more constant. You know, if you move from the bottom to the top. Notch signaling here perform two functions. One function will help wind to promote proliferation of the stem cells. You know, at the bottom of the intestinal crypt, the LGR5 plus cell and the plus 4 cell will have to proliferate to maintain the stem cell population. You need both wind and notch to make that happen. But the second function of notch is in some of these cells, in the stem cells, in the transient amplifying cells, you have less or more notch. Apparently, you have dropping wind all the way from the bottom to top. But in some of these progenitor cells, the levels of notch will be different. That will make some of these progenitor cells to become goblet cell. That is some cells that have a different function from the enterocyte. Okay, so these progenitor cells will become either goblet cell or the enterocyte. So how the progenitors make that decision, it is because they have different levels of notch in these two types of cells. So that's application of the notch pathway. So I still have two pathways to go. I will kind of accelerate and try to conclude earlier. So the fourth I'm going to explain is called TGF beta signaling. So this is kind of similar to the wind signaling in general principle, but it doesn't involve inhibition. It doesn't involve inhibition. So the TGF beta receptor have two types. One is called receptor type 2, the other is receptor type 1. One of the receptor, actually maybe both receptors, they have kinase domain. That means they can perform phosphorylation function. So once you have TGF beta-like ligands, there are tons of them. Many members of the TGF beta can become ligands of these two receptors. And we say the type is not a one receptor. You could have many, like maybe 15 proteins that belong to the receptor type 2 and another dozen that belongs to receptor type 1. So we're just trying to summarize, you know, the different family proteins to these two types. So when TGF beta ligand binds to receptor 2, it's going to recruit and cause dimerization of both receptors. You see, they become dimers. The receptor 2 become dimers, the receptor 1 become dimers. And then receptor 2 is going to phosphorylate receptor 1. And then the receptor 1 will phosphorylate a protein called SMAD. So this is called so-called regulatory SMAD. So SMAD gets phosphorylated, and then SMAD will bind to another protein called SMAD4. SMAD4 is so-called a co-SMAD. That means this SMAD is a cofactor. It is required for all the SMAD. So if you see here, we say SMAD, but in reality we have SMAD2, SMAD3, SMAD5, 6, 7, SMAD1. So all kinds of SMAD. All those other SMADs are called regulatory SMAD, while SMAD4 is called the co-SMAD. It is a common component for this pathway. So the regulatory component SMAD will have to bind to SMAD4, and then together they're going to move into the nucleus and trigger transcription. So this is a signaling transduction pathway. What is involved is a phosphorylation of the major player. Therefore, the major player can go to the nucleus and make something to happen. But here we don't see an inhibition complex involved. Well, in some of the other pathways, apparently in a lot of pathways, you do have the inhibition component in there. So here is the example how the event pathway works. But I also want to introduce to you to another principle, that is signaling and counter signaling gradients can be coordinated. Therefore, this mechanism is used to dictate self-renewal or differentiation. Here I give you an example. That is what happens in Drosophila ovary. So in Drosophila ovary, there is this place called stem cell niche. Basically, it's a place that have a special, that created a special environment. So the niche cells will release a protein which is a TGF beta family protein. Actually, two kinds of proteins. One is called such a difficult name, called decapentaplegic, or DPP. The other is called glass-bottomed boat. So that's the weird names by the scientists in developmental biology. DPP or GBB are secreted from the niche cells. They are not the stem cells. So you have the stem cell, the germ stem cells will eventually give rise to germ cells, oocyte and sperm. In this case, so the female and male reproduction cells, germ cells. That this is the stem cells of it. This is germ stem cells. So the niche cell, that's the supporting cell of the stem cell, will release this TGF beta protein called DPP and GBB. The function of them is to inhibit. It's not activation. It's an inhibition. So this is one thing I want to remind you: when a factor goes into the nucleus, although I explained in the earlier pathways I described, they always trigger gene transcription, but not necessarily. Sometimes, like in this case, when you have a GFP, when you have a TGF beta family protein that moves into the nucleus, the function actually is the inhibition of gene transcription. The gene is called BAM. Okay, so the TGF beta family protein will inhibit BAM. Therefore, you can expect that there will be a BMP or TGF gradient here, right? So because the niche cell released that BMP, DPP, GBB, therefore you can see that the gradient will become, you're going to see reduce the level when you move away from the niche. In the niche, you have high activity of DPP, GBB, but when you move away from it, you have less and less signaling in there. Okay, so therefore, in the niche, you're going to see cell proliferation because BAM is inhibited. Now, BAM is a very important protein that is released from the germ cyst cell. So the germ cyst cell is the immediate progenitor of the germ cell, like an oocyte. Okay, so this germ cyst cell will release this protein called BAM. So the BAM protein actually triggers differentiation. So the BAM is released from the germ cyst cell, while DPP-GBB is released from the stem cell. So when you get closer to the niche, you have cell proliferation. When the cell move out of that niche, and they start to differentiate because you have less GPP and DBB that inhibit BAM, and you have more BAM released from cyst cell that triggers differentiation. So this kind of signal and counter signaling gradient is frequently involved in biological processes. Okay, you have counter signaling from different sources. The very last one I'm going to introduce to you today is the receptor tyrosine kinases. I wouldn't want to say that this 5 signaling pathways, which one is more important. I won't say that because everything in life, processes are important, but this one is maybe the most widely studied. So this is called receptor tyrosine kinases. So this receptor tyrosine kinases is unique from what I have described so far. Is almost all the signaling transduction is through phosphorylation. So one adapter will phosphorylate another protein, and that protein will phosphorylate yet another protein, and eventually that signal gets transduced by the phosphorylation, followed by phosphorylation, and something at the end of that cascade, it will move into the nucleus and get some gene transcribed. So in this case, the receptor itself is called receptor tyrosine kinase, or RTK. It's because usually, they are not phosphorylated. They themselves are kinases, but they also have some tyrosine residues on it. So that the receptor have tyrosine residues, and because of the ligand binding, so you have ligand. So here we just say ligand because there are tons of them. We must refer to a very specific pathway, so I can give you the ligand. This is a family, so I say just the ligand. When the ligand binds to the receptor, that will cause phosphorylation of the tyrosine residue on that receptor. And that event will cause dimerization of the receptor, and then the kinase function of that receptor is now activated. Now that themselves get phosphorylated and they become kinases also. They get additional proteins phosphorylated, and then the additional phosphorylated protein are also kinases. So they're going to phosphorylate, in this case, like RAF, and RAF is a kinase. The RAF gets phosphorylated, but RAF itself is a kinase. Before the phosphorylation of RAF, RAF is inactivated, but now with the phosphorylation of RAF, RAF become activated. And RAF will phosphorylate MEK, Merck. Merck, same thing. It's a kinase, but it's inactivated at the very beginning. Only when it is phosphorylated become active. Now, Merck will phosphorylate ERK, ERK, and ERK will phosphorylate additional things. So this pathway, the receptor tyrosine kinase pathway, involves many phosphorylation events. It involves protein to protein interaction, because that phosphorylation won't occur if the two proteins are so far away from each other. When it's that they are enzyme and substrate, they must bind to each other to the active site to get phosphorylated. It's the cascade of phosphorylation event that will transduce the signal. And this is super important not only for stem cell research, but also for cancer research. Nowadays, a lot of drug that's very effective in curing cancer is inhibitors of receptor tyrosine kinases. Because why you want to use inhibitors for receptor tyrosine kinases? Because remember that domain is it's outside of the cell. So that inhibitor, if you have developed an inhibitor, that inhibitor doesn't have to penetrate the cell, which is not an easy task. You have an inhibitor, that inhibitor goes into the cell to perform a function. That is a lot more difficult if you have the choice to use an inhibitor to block the extracellular domain. That's why there are a lot of drugs these days that are designed to attack the ligand receptor, the receptor tyrosine kinases. Therefore, you can block the pathway. That being said, there are also other drugs that are small molecules. They can enter the cell and block RAF, MEK, and ERK. They're all inhibitors developed, especially in the treatment of melanoma and breast cancer. And now it gets expanded to lung cancer and other cancers as well. So this pathway, as you will learn, I hope you learn something in this lecture, but in the future, most likely you're going to encounter the usage of this pathway in your career, no matter you're going to join a pharmaceutical company, or if you're going to pursue a medical degree and practice medicine. So it's highly likely you're going to encounter the drugs, attacking this pathway. So there are about more than 20 classes of receptor tyrosine kinases. So here we listed the 20 classes. 20 classes. In each class, you have many proteins involved. So I just want to point out the family 4, that's EGF family, family 3, that's PDGF receptor family, and family 5, that's FGF receptor family. So these 3 families are widely studied in stem cells. I won't say that they are the most important in stem cell research, because sometimes we don't know something, we haven't studied it. It doesn't mean it is not important. For example, some of those red ROS, maybe we didn't have a lot of resources there in study their function in stem cells. But I would say that EGF receptor, FGF receptor, and PDGF receptor families are widely studied in stem cell research. So again, I want you to appreciate the general principle and try to memorize some of the key components of each pathway, but try not to become confused. And then once we encounter that again, you can add to your knowledge. Okay, that's the end of today's lecture. And if you have questions, you can stay behind, or otherwise you can leave. Thank you.